Alright, so we're going to pick up where we left off with the physiology of a neuron. Like I said before, the neuron membranes are designed and specialized for communication. The messages that the nerves send to one another are called nerve impulses or action potentials. Basically, this is an electrochemical signal caused by sodium ions and potassium ions moving across the plasma membrane. So, here's my question for you. What are ions? Thinking all the way back to chapter 2, what is the definition of an ion? Yeah, very good. I don't really know if you answered it, but I'm going to assume that you did. An ion, sorry for that weird noise, I'm putting on some lotion because it's very dry in here. Sorry about that. So an ion is going to be a charged atom. Anything that has a charge. Could be a positive charge, could be a negative charge, but it's going to be a charged atom. It has either too many or too few electrons. So a sodium ion would be a positively charged sodium atom. Potassium ion is a positively charged potassium atom. Are ions, so here's another question for you, if you can think all the way back to chapters 2 and 3, are ions able to pass through the plasma membrane on their own, or do they require the aid of protein carriers and channels? All right, thinking back to our hydrophilic, hydrophobic environments, are we going to need to move our atoms, or can they diffuse through the plasma membrane, or do they require special transporters? They are going to require special transporters. If you remember, the plasma membrane keeps out anything that is too large or too charged. Charged ions, like water. Water itself is charged, has all those partial charges. Remember when we talked about how water is a polar molecule, partially positive on one end, partially negative on the other end. Because of those partial charges, ions love water. They really do. They love hanging out along all of those charges. So they are not going to readily cross the plasma membrane, which is a large nonpolar region right in the middle. They're not going to cross it. They don't want to go anywhere near it. They want to hang out in the water. So in order to cross it, they're going to need special channels that are going to allow for the movement of those ions to flow across that plasma membrane. So the transport of those ions is going to be down the concentration gradient. So although we require help, do we require ATP? Do we require energy to pass through the plasma membrane if we are moving down our concentration gradient? No. As long as we're moving down our concentration gradient, and as long as we're moving downriver, we might need the aid of a boat. If we want to, float down the river, we might need the aid of a boat to get us there, but we don't require any energy as long as we're flowing with the current. As long as we're moving down our concentration gradient, our ions do not need ATP to help get them where they need to go. So, when we're going to use that movement, we're going to use those concentration gradients to help us conduct our nerve impulses. So when a signal travels down the length of the axon in the cell, it's going to need to pass the message on to the next cell in the pathway. Now these cells are going to be designed for really quick and efficient communication, but we still need to get the message from inside of one cell to the inside of another cell. We do this through the release of neurotransmitters. Neurotransmitters are chemicals that diffuse across the gap between cells to convey that message. So here we have an axon, here we have the axon terminal, here we have another cell, or where we're going to be receiving that information. The space between them is what's known as the synapse. That's going to be the location or the junction between one neurocell or one neuron and another. Neurotransmitters are going to be those chemicals that diffuse across that gap. They're going to move from one cell to another cell to convey that message. The space, like I said, that space in between is called the synapse. So the synapse is going to be the region in between, the junction between one cell and another. Now the actual space in between is what's known as the synaptic cleft. This is a small gap that separates the sending neuron from the receiving neuron. And that neurotransmitter is going to transmit the action potential across that synapse. It's going to be stored inside of these little synaptic vesicles at the axon terminal. Those synaptic vesicles So that'll be this little vesicle right here. It's going to bind to the synaptic membrane, or it's going to bind to that membrane at the synapse, and it's going to dump. It's going to release all those neurotransmitters are going to dump into the receiving neuron where they can pass along through those ion channels. So here's what's occurring. Here's the events that are occurring at the synapse. This nerve signal is going to travel along the axon until it reaches the axon terminal, the ending point of that axon, this region right here. Calcium ions are going to enter the terminal and stimulate the synaptic vessels to merge with the membrane. So all these little synaptic vessels that are here, an influx of calcium ions is going to encourage these synaptic vesicles to merge with the membrane and dump. They're just going to start dumping massive amounts of neurotransmitters. That neurotransmitter is going to be released into the synaptic cleft. That'll be this region right here. And it's going to diffuse across to the receiving membrane. Once we diffuse across into the receiving membrane, they're going to bind to receptor proteins. Once we bind to receptor proteins, that information is going to be transmitted along the length of the axon and passed on to the next cell in the process. So there are a variety of different types of neurotransmitter, and each neurotransmitter plays a very different role in the production of different effects on different types of cells. So for example, acetylcholine triggers the skeletal muscle contractions in both the peripheral and central nervous system. If we were to lose the receptors responsible for receiving the acetylcholine receptors, what would happen? Okay, so let me give that information to you again. Acetylcholine triggers skeletal muscle contractions. So we're triggering the contraction of our skeletal muscles in the peripheral nervous system and the central nervous system. What would happen if we were to lose the receptors responsible for receiving the acetylcholine receptors? What would happen? So just an example, this is a real life homeostatic imbalance. Myasthenia gravis is an autoimmune disease that attacks the acetylcholine receptors at the neuromuscular junctions. So it results ultimately in very little muscular strength and loss of control over voluntary muscle movement. You can send those signals as much as you want. So it's unlike ALS where we're actually destroying the motor neurons. We're not destroying the neurons. The neurons are perfectly intact. We are sending the information. We're sending the acetylcholine neurotransmitter. The problem is that the muscles aren't receiving the message. So it's kind of like a child plugging their ears and going, la, 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 I can't hear you. No matter how loud you shout, that child still can't hear you. That's what's happening. No matter how much acetylcholine you send, without the receptors, they cannot receive that information and they cannot then trigger muscle response. Pardon me. So the end result is going to be very little muscle strength or a loss of control over that voluntary muscle movement. There are actually a lot of different neurotransmitters that are going to have a variety of different effects. So for example, depression. Depression is caused by a decrease in serotonin. Serotonin is a very important neurotransmitter. Depression can also be caused by a decrease in norepinephrine or dopamine. Again, all of these are various neurotransmitters. Parkinson's disease is caused by decreases in dopamine. ADHD is caused by levels of norepinephrine and dopamine can contribute to ADHD. So that is going to be the sort of the significance of neurotransmitters. We do want to talk just very briefly about the removal of a neurotransmitter. So this would be, we've talked a little bit about excitation and how if the neurotransmitter causes sodium gates to open, it's going to excite that neuron, it's going to pass that information on. Inhibition occurs if a neurotransmitter causes potassium ions to exit the receiving neuron. So this is going to be after the neurotransmitter has initiated the response, it's going to be removed from the synaptic cleft. Some synapses have enzymes that inactivate that neurotransmitter. So for example, the enzyme acetylcholinesterase breaks down acetylcholine after the message has been carried out. So in other synapses, the sending membrane can actually just reabsorb the excess neurotransmitter once it's sent that message. The short existence of neurotransmitters at a synapse prevents the continuous stimulation of the receiving membranes. So we excite and then we inhibit. We don't want to continually stimulate that nerve over and over and over again. If the neurotransmitters were to linger in the cleft, the receiving cell would be unable to respond to a new signal from a different cell. So it would be unable to respond to any other information being sent if that previous neurotransmitter is still there. So like I said, there are a lot of different neurotransmitters. There are more than 100 different types: acetylcholine, norepinephrine, dopamine, serotonin, glutamate, GABA. All of these are different types of neurotransmitters. These neurotransmitters are going to vary depending on what it is that we are trying to stimulate. If it's nerve to muscle, nerve to organ, nerve to gland synapses, they're all going to use neurotransmitters, and they're all going to use different types of neurotransmitters depending on what is being called for. Acetylcholine and norepinephrine are going to be active in both the central nervous and the peripheral nervous system. In the peripheral nervous system, they're going to act at the neuromuscular junction. So they're going to be responsible for this nerve to muscle connection. In the peripheral nervous system, acetylcholine is going to excite skeletal muscle, but it's also going to inhibit cardiac muscle. So it's going to have a very important role depending on what it's being used for and where it's being located, depending on just where it's going. Norepinephrine is going to excite smooth muscle. In the central nervous system, however, norepinephrine is going to play a very important role in dreaming, waking, and mood. Serotonin is going to be involved in thermoregulation, sleeping, emotions, and perception. Many drugs act at the synapse. So a lot of drugs that are used to help with neurotransmitter imbalances, a lot of times they're functioning at that synapse. They're actually functioning to either increase or decrease the absorption of those neurotransmitter molecules. Some can even act in the increase or decrease of the release of those neurotransmitters, but their primary goal is going to be to slow down those neurotransmitters to prevent the overstimulation or understimulation of the nerves at that location. So here's an example of a single nerve. See how it's receiving messages from multiple nerves, from multiple locations. This is why it's very important to not continuously stimulate with the presence of neurotransmitters. This is why it's so important to absorb the excess or remove the excess, inhibit that response, because if we're receiving if we're receiving a message from this neuron here, we cannot then receive or respond to a message from any of the other neurons that this cell is communicating with. So it's very important to clean up the excess neurotransmitters once the signal has been sent. All right, you should be able to describe the 3 types of neurons, list the 3 main parts of a neuron. You should also be able to describe how nerve impulse is propagated and summarize how that nerve impulse is transmitted from one neuron to the next. So be able to talk about what's happening at that synapse and at that synaptic cleft. Please let me know if you have any questions, comments, or concerns.